Cell structure and membrane transport. In this lecture, you'll learn about the 8 characteristics of life that define living things. You'll be introduced to the prokaryotic cells and their structural components, and I'll talk a little bit about eukaryotic cells and the differences and similarities between them and prokaryotic cells. Lastly, I'll discuss the transport of materials across the cell membrane. There are 8 major characteristics that define living things. At the center of this list is cellular organization, which means that the cell is the basic unit of life. Growth and development. Organisms can grow at the individual level, for example, growth in size, and also at the population level, growth in numbers. Development refers to maturation. For example, as humans, we mature into adults from children. There are also types of cells that mature into fully functional cells. We'll learn more about that later. All living things have the ability to respond to internal and external stimuli. For example, humans snatch their hand away when they touch something hot. Cells swim towards nutrients, or they may swim away from toxic chemicals. The ability to transport molecules across the cell membrane into and out of the cells, as well as transporting materials throughout a multicellular organism, as we transport oxygen through our bodies. Nutrients transported into cells are then metabolized by the use of enzymes. Living organisms are also able to reproduce, produce offspring. And by doing so, they pass on genetic traits to their offspring through their DNA. So what about viruses? The only characteristics viruses have is heredity, specifically DNA or RNA, but not both. Viruses are not made of cells, nor can they do any of the things listed here that cells can do. In the first lecture, you learned that microbes can be made of prokaryotic or eukaryotic cells, and all cells can be categorized into these 2 large groups. Bacteria and archaea are prokaryotic organisms, and all other organisms consist of 1 or more eukaryotic cells. Similarities among the 2 cell types are that both have a cytoplasm contained within a cell membrane. Both contain genetic information in the form of 1 or more chromosomes, and all cells contain ribosomes for the purpose of protein production. There are many more differences than similarities between the 2 cell types, and you should keep track of those differences as we go along. Let's take a closer look at the structure of a prokaryotic bacterial cell. All of the structures starred in red on the right are found in the cytoplasm. Keep in mind that this is a composite cell, which means all of the structures here can be found in prokaryotic cells, but an individual prokaryotic species may only have some of these structures. For example, not all bacteria can swim, so those microbes that can't swim would not have a flagellum. I'll discuss each of these structures separately, starting on the outside of the cell, and then we'll work our way into the cytoplasm. The cell envelope is the external covering outside of the cytoplasm, and it's composed of 2 or 3 layers. The number of layers can be determined by a special stain called a Gram stain. The cell envelope is comprised of the cell membrane that is just outside of the cytoplasm. This is the first layer of the cell envelope, and all organisms have a cell membrane, also known as a plasma membrane or cytoplasmic membrane. All bacteria have a cell wall, which is just outside of the cell membrane and consists of 1 layer or 2. Bacteria can be classified into one of 2 groups based on their reaction to the Gram stain. The reaction to the Gram stain gives an idea of the structure of a cell's envelope. Organisms that stain purple after the Gram stain are considered gram-positive, and their cell envelopes are 2 layers thick. And those that stain red after the Gram stain procedure are gram-negative bacteria and have 3 layers to their envelope. Again, the cell envelope includes the plasma membrane and the cell wall. On the left of this figure is the cell envelope of a gram-positive organism, like Staphylococcus aureus, and on the right is the cell envelope of a gram-negative bacterium, like Escherichia coli or E. coli. In both groups of bacteria, there is a layer of peptidoglycan outside of the cell or the plasma membrane. In gram-positive organisms, this layer of peptidoglycan is very thick and is the only layer of the cell wall. In gram-negative organisms, this layer of peptidoglycan is very thin, and there is also another layer called the outer membrane. So the cell wall of gram-negative bacteria has 2 layers. In between the plasma membrane and the layer outside of that is the periplasmic space. This space is more extensive in gram-negative organisms because of the additional outer membrane. This space consists of a gel-like matrix and contains proteins, enzymes, and ions, among other molecules. You should be able to explain the differences between the cell envelope versus the cell wall and differentiate between the cell wall of a gram-positive versus a gram-negative bacterium. The cell wall is responsible for maintaining cell shape and the stability of the cell, protecting them from osmotic changes in the environment. Peptidoglycan is the primary component of the cell wall in both gram-positive and gram-negative organisms, but much more extensive in gram-positive organisms. Peptidoglycan is made up of the sugars N-acetylglucosamine and N-acetylmuramic acid, abbreviated as NAG and NAM, respectively. And it also consists of proteins. The NAG and NAM sugars differ in how they are linked in gram-positive versus gram-negative organisms. In gram-negative bacteria, the NAG and NAM are linked by short peptide or proteins, but there is a direct link between the sugars in the gram-negative bacteria. The cell wall of gram-negative bacteria is made up of a thin layer of peptidoglycan and an outer membrane. The outer membrane consists of a bilayer of phospholipids just like the cell membrane. Lipopolysaccharides are embedded in the outer membrane. Lipo stands for the lipid A molecule, and polysaccharide means many sugars. This is an example of an LPS molecule. So these molecules have a lipid portion that anchor the molecule to the outer membrane, and then there are these many sugars attached to the lipid that hang on the outside of the cell like a flag. Lipopolysaccharides, or LPS molecules, are considered endotoxins during infections with gram-negative bacteria. These LPS molecules can cause symptoms like fever and septic shock. The O antigen differs among gram-negative pathogenic bacteria and is how they are classified. In E. coli 0157H7, for example, the O stands for the O antigen, where O is the abbreviation for oligosaccharide, which is just another name for polysaccharide. You'll also find other elements embedded in the outer membrane, such as the porin proteins that allow molecules to be transported into and out of the cell. Again, there is a thin layer of peptidoglycan in gram-negative bacteria and a more extensive periplasmic space, which is where the metabolic reactions occur. The gram-positive cell walls consist of a thick layer of peptidoglycan only. There is no outer membrane, and instead of LPS, gram-positive bacteria have teichoic acid and lipoteichoic acid. The lipoteichoic acid is not pictured here. The lipo or lipid portion of the lipoteichoic acid anchors the molecule to the cell membrane. In the teichoic acid is embedded just inside the surface of the peptidoglycan layer and does not extend all the way to the plasma membrane. Both acids serve in cell wall maintenance and help maintain the rigidness of the peptidoglycan cell wall. Inside of a host, these acids increase the ability of these organisms to cause infection. Just like in gram-negative bacteria, in the periplasmic space, there are enzymes, proteins, and other molecules, and it is the site of metabolic reactions. Some gram-positive bacteria, such as Mycobacterium tuberculosis, that causes TB, have another layer outside of the peptidoglycan layer made up of mycolic acid, which makes the bacteria very difficult to stain. It also makes these cells very, very sticky, and they're very slow growing. The acid-fast stain contains acid and strong dyes to penetrate the waxy mycolic acid layer. These organisms stain red after the acid-fast stain is complete. This table is a nice overview of the differences between gram-positive and gram-negative cell walls. We've discussed all of these differences, but I want to point out the permeability characteristic. Due to the outer membrane, gram-negative organisms are less penetrable by molecules, for example antibiotics. These molecules don't pass the outer membrane as easily as they pass the thick layer of peptidoglycan in gram-positive organisms. Therefore, gram-positive organisms are generally more susceptible to antibiotics and other chemicals than gram-negative organisms. The gram stain allows you to group organisms into two broad categories of gram-positive and gram-negative. This stain was created by Hans Christian Gram, which is why Gram is always capitalized. It's an important first step in the classification and identification of unknown bacteria. The Gram stain also aids in diagnosing an infection and guiding drug treatment based on how they affect gram-positive or gram-negative cells. For example, penicillin acts very well on the cell wall to kill gram-positive bacteria, but gram-negative bacteria are much more resistant to penicillin because they have an outer membrane that protects their peptidoglycan layer. The Gram stain is a differential stain, which means it allows you to tell the difference between two different groups of cells that are based on the makeup of their cell wall. There are other types of differential stains, like the acid-fast stain that I just mentioned, that allows you to differentiate two groups of organisms based on other differences in their structures. This chart shows us each step of the Gram stain procedure, the chemicals that are added to the cells, and the appearance of the gram-positive and gram-negative cells after each step. If we have a mixture of gram-positive and gram-negative cells on a slide, we'll be able to see both types of cells at the end of this staining procedure. In step 1, shown here in the first row, crystal violet is added to the slide. The crystal violet is the primary stain, and in all differential type stains, by the end of the process, the cells that take on the color of the primary stain are positive for that stain. So cells that take on the crystal violet and remain purple at the end of the gram at the gram stain are considered positive for that stain, or gram positive. After step 1, both gram positive and gram negative cells are purple. You see both of these cells, gram positive and gram negative, are purple. Step 2 is application of the mordant. A mordant is a chemical or procedure that allows the primary stain to bind more tightly to the cells. In the gram stain, Gram's iodine is the mordant. When added to the cells, the Gram's iodine forms a complex with the crystal violet, and this causes the stain to be trapped in the thick peptidoglycan layer of the gram-positive cells. After step 2, both gram-positive and gram-negative cells are still purple. What makes this a differential stain and allows us to see the two types of cells on one slide is the decolorizer step. This step removes the stain from those cells that are negative for the differential stain. In this case of the gram stain, alcohol is added to the slide. The crystal violet Gram's iodine complex remains trapped in the thick layer of peptidoglycan, but since the gram-negative cells have such a thin layer of peptidoglycan, the outer membrane becomes leaky, and the crystal violet is easily washed out of the thin layer of peptidoglycan, leaving the gram-negative cells colorless. In order to visualize the gram-negative cells, the slide is then covered in safranin. But because the crystal violet is so much darker than the safranin, this has no effect on the gram-positive cells, and they will remain purple. But the colorless gram-negative cells are stained by the safranin, and they are now stained red or pink at the end of the procedure, and we can visualize both gram-positive and gram-negative cells on one slide. Other structures located on the outside of cells are appendages and glycocalyces. Flagella are appendages that allow organisms to move, and they're located on the outside of the cell. Axial filaments, also known as periplasmic flagella, are located within the periplasmic space, but also allow movement. Appendages that allow bacterial cells to attach to other cells or surfaces are fimbriae and pili. Fimbriae are short, bristle-like structures that allow cells to attach to surfaces and other cells, which is important during the infectious process. As seen in this figure here in the center, there are E. coli cells using fimbriae to attach to intestinal cells. Pili, which is plural for pilus, are longer and thicker than fimbriae. There are different types of pili, but one type in particular is the sex pilus that allows transfer of DNA from one cell to another, as seen in this figure here. We see the pilus connecting these two cells so that they can easily transfer DNA, like the plasmids I mentioned. Flagella allow bacterial cells to swim, and they are composed of a filament, a hook, and a basal body. The filament, shown here in green, is a long, thin helical structure made of flagellin protein, which is considered the H antigen. In the pathogenic strain of E. coli, O15787, the strain is classified by the type of oligosaccharide, or LPS, as mentioned earlier, which is O157, and the type of flagellin, which in this case is H7. Different strains of E. coli are characterized by the differences in their LPS, flagellin, or other cellular structures. You can read this short article that discusses serotyping Salmonella to get a better understanding of this concept. The hook is a curved sheath shown in light blue that protects and surrounds the filament. The basal body is a stack of rings that are firmly anchored to the cell wall. In gram-negative organisms, the rings are attached to the cell membrane and the outer membrane, and in the gram-positive cells, the rings of the basal body are attached to the cell membrane. Bacteria have varied arrangements of flagella. Monotrichus is a single flagella. Amphitrichus is a single flagella at opposite ends of the cell. Lophotrichus is a bunch of flagella at one end of the cell, and peritrichous flagella are those that cover the entire surface of the cell. Flagella help bacteria move in response to environmental stimuli such as light and chemicals. Flagella allow bacteria to move towards or away from a stimulus, and this is termed taxis. We can further define taxis based on the stimulus the bacteria are moving towards or away from. Positive taxis is when an organism is moving towards something, and negative taxis is when it's moving away from something. For example, chemotaxis is when a bacterial cell is moving towards or away from a chemical stimuli. Phototaxis is movement towards or away from light. There are microbes called cyanobacteria that are able to undergo photosynthesis just like plants and will move towards light via phototaxis. It takes a lot of energy to move the flagella, so bacteria don't expend much energy moving their flagella unless they have a reason. When they do move toward a stimulus, let's say some type of a nutrient, the cell moves in runs and tumbles, as we see here. A run is when the flagella is spinning in a counterclockwise direction, which allows the cell to move straight. After a few seconds, the flagella moves in the opposite clockwise direction, and the cell appears to fall. The flagella repeat these sequences until the cell reaches its destination, kind of like the fluttering of a butterfly. They don't really look like they have a final destination by watching the movement of the runs and tumbles, but the net direction of the movement, as pictured here on the right in this figure, is in one direction, towards the attractant. On the left side of this figure is a bacterial cell that is just moving around in one area because there is no attractant or repellent to get that bacteria to try and move in any one direction. There is nothing attracting or repelling it. The glycocalyx is outside of the cell wall, and it's made of sugars and or proteins. The glycocalyx can be loosely attached to the cell, as in the slime layer, or tightly attached and highly organized, like in the capsule. Glycocalyx provides the bacterial cells with protection from dehydration, nutrient loss, allows bacteria to attach to surfaces, and it also aids in biofilm formation. Biofilms are structures that consist of layers of bacteria stuck together on a surface. These biofilms provide lots of protections for the organisms that are contained within them, and they can be very troublesome. For example, bacterium biofilms are more resistant to the effects of antibiotics, which make it a challenge to treat infections. Capsules help pathogenic bacteria avoid phagocytosis or destruction by human immune cells. They're also very difficult to stain, So a negative stain is often used, which stains the background, leaving the cells unstained, as seen in the figure on the right here, where you see the dye, the dark dye in the background of the negative stain, and then you see these white halos of bacterial cells. The cytoplasm is a dense gelatinous solution that contains all of those biomolecules we discussed: carbohydrates, proteins, lipids, and nucleic acids, among other things that function in cell synthesis and metabolites for the cell to produce energy. The cytoplasm is about 70 to 80% water, and this percentage depends on the cell's external environment. The nucleoid is an area inside the cell, as pictured here, that contains the bacterial chromosome and the nucleoid-associated proteins that help in organization and packing the chromosome into the cell. There is one single circular chromosome. It's circular but coiled tightly so that it fits neatly inside of the cell. It is also a double-stranded DNA molecule. It's not surrounded by a membrane like in eukaryotic cells that have a nuclear membrane. The chromosome contains all of the genes that are necessary for the growth and development of the organism. Plasmids are extra pieces of DNA. As seen in this figure here at the bottom, we can see the nucleoid that contains a single circular chromosome, and then this small circle here, which is the plasmid. So again, plasmids are extra pieces of DNA. They're also circular and double-stranded, but they are much, much smaller than the chromosome. They contain non-essential genes, and as mentioned before, they confer special properties onto the organisms, like antibiotic resistance. Plasmids are used in genetic engineering because of their small size. They are very easy to manipulate and quickly transferred from cell to cell. Plasmids are not associated with the nucleoid. You see, they are outside of the nucleoid. The bacterial ribosome is also inside of the cytoplasm and is composed of ribosomal RNA and protein, and it's the organelle that produces proteins. The ribosome consists of a large and a small subunit that come together to make the complete ribosome. Prokaryotic ribosomes are much smaller than eukaryotic ribosomes. Prokaryotic ribosomes are 70S, where the large subunit is 50S and the small subunit is 30S. But 50 and 30 doesn't equal 70 because the S stands for Svedberg units and is not based on mass, but is based on the sedimentation rate, how fast something settles to the bottom in a solution. Eukaryotic ribosomes are 80S, which means they sediment faster than the 70S prokaryotic ribosome. Ribosomes in archaea are also 70S but have different proteins and ribosomal RNA molecules. Ribosomes are found in all cells because all cells require proteins. Inclusion bodies and storage granules may also be found in the cytoplasm of some bacterial species. These structures store nutrients and other substances. They vary in size, number, and content depending on the cell. Pictured on the left is a bacterium that contains many sulfur granules in its cytoplasm, and on the right is a bacterium that contains inclusion bodies that have little magnets. These magnets help orient the cell in its aquatic environment. Bacteria can use nutrients stored in granules when environmental sources are depleted. Just like we have a skeleton, prokaryotes and eukaryotes also have a cytoskeleton. It was previously thought that prokaryotes did not have a cytoskeleton, but in this figure pictured here, you can see that they do. These bacterial cells are stained with a green fluorescent dye and viewed with the fluorescent microscope. We can see that the cytoskeleton of this organism is shaped like a helix. The cytoskeleton is important for maintenance of cell shape and structure. It functions to maintain cell integrity during cell division and allows the cell to move and squeeze through small spaces without collapsing. Bacterial cells vary in shape, size, and arrangements and are typically described by one of three basic shapes. Repeat after me: coccus or cocci for plural. This is a round or spherical shaped cell. Bacillus or bacilli, plural, is a rod-shaped organism. Shapes of some organisms are not so easy to determine and may be a variation. For example, coccobacillus are short rods and are sometimes difficult to make out as rods and can sometimes be mistaken for cocci. There are also bacilli that are gently curved like a comma, and these are called vibrio. A little more extreme are the spirillum and the spirochetes that are spiral or corkscrew shaped. Bacteria also differ in their cell arrangement based on how they divide. In the table on the left is a list of cell arrangements, and on the right are the planes of division of those cell arrangements, or location of where the cell splits in half during cell division. Cell shape and cell arrangement are the first characteristics considered during bacterial identification. You don't have to memorize the cell arrangements, but you should know that the arrangements are due to the different planes of cell division. Organisms that belong to the archaea domain are very interesting and unique. These microbes are more closely related genetically to eukarya than they are to bacteria due to unique genetic sequences in their ribosomal RNA, even though they look like bacterial cells. Organisms that belong to the archaea domain have unique membrane lipids and unique cell walls that allow them to live in the most extreme habitats in nature, which is why they are often referred to as extremophiles, organisms that love extreme conditions. These organisms are grouped according to their habitats. Those that prefer extreme heat are hyperthermophiles. If they prefer very acidic conditions, they are acidophiles. Extreme cold, they are psychrophiles. Pictured here are salt evaporation ponds. These are man-made structures where salt is produced. The halophiles produce pigment and gradually shift the color of the ponds to pink, red, and orange. How cool is that? You should become very familiar with this Venn diagram, which lists characteristics unique to prokaryotes versus eukaryotes, as well as those that are common to both types of cells. You should refer back to this slide when learning about eukaryotic cells. This is the end of part 1. Thank you for listening.